Good morning, everyone. I am Maria Adelaida Gomez. I'm an investigator here at CIDEIM, and I will be with you this semester coordinating the course on pathobiology of intracellular infections. I am very happy to tell you that for this course we have 122 participants from many different institutions within Colombia and also in the Americas, and we have expanded this year as well to Europe. So hello, our colleagues from Spain and Italy, and also Central America as well, Mexico, North America. So we have participants from Armenia, Bogotá, Bucaramanga, Cali, Cartagena, Medellín, Montería, Pamplona, Pereira, Santa Marta, Sincelejo, Villeta, Yopal. Hello, everyone from Colombia as well. Chile, Guatemala, Honduras, Mexico, Peru, and Venezuela. España and Italy, I have already mentioned you, so everyone, very welcome. Today, we have a very special guest. We have Dr. Kristen Schutte. Dr. Schutte is the co-PI of the CIDEIM Yale Fogarty program on global infectious diseases, and this is the program that allows us to do these courses. And he's going to give the opening talk for this semester on microbial pathogenesis. Dr. Schutte is the John Rotman Paul Professor of Epidemiology at Yale University. He is also the Director of Graduate Studies at the School of Public Health, also at Yale University. His research program is primarily on RNA biology and the RNA world of trypanosomatid parasites. He's going to tell us a little bit more about himself, and then he's going to proceed with his talk. So Dr. Schutte, thank you so much for being here. We're very happy to host you in CIDEIM. And again, welcome to everyone for this course. Let me just give you a couple of things regarding the logistics of the conference. So we're going to have the talk in English, as you all know. We have activated a chat session for all of you that are taking the course on the web base. So if you have any questions, you can communicate via chat. You can also request a space to ask the question verbally. If you want to do your question in Spanish, do ask your question in Spanish and we will be happy to translate as well. So, so go ahead and enjoy the lecture.
Okay, can you all hear me? Yes? See, I have to start. Thank you very much for having me here and having the honor to doing this opening lecture. My Spanish is non-existing, so I apologize for that, but I'll do my best. So I would like to tell you a little bit more about myself, and especially I get asked a lot of times, how did I get where I'm at the moment? How did I end up being a professor at Yale University? And also along the way, how did I choose graduate school? How did I choose what research topic I want to work on? And probably the major theme that's behind it is to have good mentors. And mentors is plural, not just one. Particularly if you do your dissertation or your master research and you have a faculty member assigned to you, that's certainly a great mentor. But there are lots of times, occasions where you can't really talk to that mentor about some of your problems or questions you have because you might not feel comfortable. So it's actually good to have several mentors that can guide you through your life. And that has helped me all along from the very early on to where I'm now. And I have to say, until I became full professor at Yale, I always had mentors that would guide me through and tell me, don't do that. That was actually the major thing that I learned. And I'm doing this now to junior faculty and students. Most of the time I tell them, don't do this, don't do that. That's too diverse. You have to focus. So I hope this helps a little bit. So I'm now a full professor at Yale. And that's always challenging, of course, as well, but it's a lot of fun doing research. I enjoy it very much. I wouldn't be doing this if I wouldn't enjoy it, and I think the faculty here will probably agree with the same. It's like I could easily retire now. So I was asked to give an overview of this course, which I find a fascinating course, I have to say. But also I think it's going to be a challenging course. And if this I worked a minute ago. Yep. So, as you all know, this is a course on the pathobiology of intracellular infections. And the objective of this course is to provide the knowledge to develop understanding of the complex molecular processes instigated by intracellular microbial pathogens in the establishment of infections and induction of disease, and to apply these concepts to design of therapeutic and prophylactic strategies. That is a lot. That is an enormous amount of topic to cover. But as I said, it's going to be interesting, it's going to be fascinating, but I think it's also going to be challenging. And so what I've shown down here are the three classes of organisms that you're going to discuss during this course. Viruses, as shown as the Ebola virus, then bacteria, intracellular bacteria, shown here for Salmonella, and then parasites, which is illustrated here by Plasmodium. Different stages, and this already should give you a very good idea that we're going to talk about very diverse group of organisms that first of all look very different, and they obviously, since they look very different, they're also going to do things very, very differently. And so they're going to be lectures covering all these three organisms, trying to get you familiar with how they enter cells, how they cause disease, and how we can actually defeat them. This obviously needs a little bit of background because you're going to be talking and reviewing papers from lecturers that will come through. And I've just selected 3 of them here from 3 lectures that will be talking. We're on Crothers. And just to give you a little flavor of what you're going to be dealing with is the rapid membrane disruption by perforin-like protein facilitates parasite exit from host cells. Then Craig Roy from Yale works on Legionella, and he has a paper recently. Effector inhibits host autophagy through irreversible ATG8 deconjunction. And then Walter Mothers, also from Yale, is a virologist. He had a paper on retroviruses use CD169-mediated transinfection of permissive lymphocytes to establish infection. I obviously do not expect all of you to understand this, and even I have troubles understanding all the titles, what all this means, but I just want to sort of expose you to the complexity that is involved in looking at this different organism, establishing intracellular infection, and then also trying to find strategies to defeat these organisms. So when I was thinking about what I should do today, I decided that I can't give you an introduction to general microbial pathogenesis. I probably could give you a book or several books that you can read, but I'm not sure that will really help. So what I thought might be helpful, and which I always like to do, is go back in history and expose you to some of the historical things that have happened in this field. And these are obviously selected, but significant historical events in microbial pathogenesis. I have to say personally, I always learn something when I look back at the history of an organism or a disease, and there's always something I missed, and something that I can learn from what people have done 20 years ago, 100 years ago, 200 years ago. So I decided to do that. We'll spend most of the time talk about historical events in microbial pathogenesis. And then as a way of introduction, talk about general concepts of microbial pathogenesis. Just for some of you, remind you of some words and terms we use in this field. For others, they're probably going to be new, but just sort of go over it and sort of introduce you to some terminology that we use in this field. Okay? Am I talking too fast? Because I was asked to not to speak too fast, so is it okay? Is it okay here?
Yeah, so far.
Just want to make sure that.
Yeah. Okay.
Yeah?
Okay.
Okay. So selected significant historical events in microbial pathogenesis. Lesson number 1. Pathogens, you probably all know this, pathogens in infectious diseases have been with us for a very, very long time. This is not something that happened yesterday. We have co-evolved with these pathogens over thousands and thousands of years. And this is from 3700 BC, and it's the first written record of a virus infection in an ancient Egyptian writing from Memphis. And if we look over here, there's a person here with 2 legs, and then this bottom leg doesn't look really normal. Some deformation here. And this looks like clinical signs of paralytic poliomyelitis. And this is polio. And it is similar what is shown in the next picture down here. This is the Pharaoh Siptah, who ruled in Egypt around this time, and he died suddenly at the age of about 20. And his mummified body actually laid undisturbed in this tomb in the Valley of the Kings until it was excavated in 1905. And again, at the left leg here, you see this very deformed foot. And this is again a typical sign of polio. So this king probably died of polio. Plagues. They have been around for a very long time. And this is the earliest recording I could find. And this is the plague of Athens, 430 BC. And this person, I'm not even going to try to pronounce this, described it as a rapid onset, raging fever, extreme thirst, bloody tongue and throat, red skin followed by pustules and blisters. And this is all the signs that we normally see in scarlet fever, which is a disease that can occur as a result of a group A streptococcus infection. So also bacterial infection, they have been around for a very, very long time. And this killed about half of Athens' population. And they just happened to be in war. They have been in war a lot of times, in the old Greek, with Sparta, and because of this impact, Athens became stagnant, and this war then continued for another 27 years. So this is something I honestly have to admit I wasn't aware of. And this is the spontaneous generation of life debate. Who is aware of this debate? Nancy?
Yeah, I read it in Biology 101.
Probably there I came across it as well.
Yeah.
But as I said, this is another one of those fascinating things when you go back in history and other things. So this is the belief in the spontaneous generation of life from non-living matter. And this was introduced by Aristotle down here, who lived around 350 BC. Now, if you read this, according to him, it was readily observable that aphids arise from the rainfall, which falls on plants, fleas from rotten matter, mice from dirty fermented corn. I always thought he was an intelligent person, but this doesn't sound too intelligent. So this actually stayed around for another 2,000 years, this belief that this happens. Now we'll come back to this. We also had pandemics. You're probably aware of them, some of them, most often referred to as the Black Death or the Dance of Death, which is a picture here by Hans Holbein around 1519. And the plague was believed to be cause of Black Death that swept through Asia and so on, 14th century, and killed an estimated 50 million people. Because all these diseases I mentioned, or these tragedies I mentioned so far, it was at that time not known what caused these diseases. This one, probably Yersinia pestis, that caused this, because that's called Black Death. And there's some interesting, statements here. Each started in coastal cities and moved inland. Those who cared for the sick were at no greater risk than those who did not. And a higher incidence of pneumonic plague occurred at the end. And of course, agricultural production dropped, leading to mass starvation during that time. And this is, we're moving now into the 15th, 16th century. This is 1546. This fellow proposed the theory of contagious diseases. He believed that diseases were spread through contact between individuals. And he developed this theory while treating cases of syphilis. If you think nowadays, this is common knowledge, this is nothing outrageous, but I think at that time coming up with this and postulating this, and this, as I said, was in 1546. This I wasn't aware, because on the next slide or so, I always thought the Dutch person was the inventor of the microscope, but there's actually this Johannes and Zacharias Janssen invented the first compound microscope in 1590. Had two sets of lenses, and they could have a magnification of 10 to 100 times in the object's actual size. Very nice. Okay, the debate of the spontaneous generation of life continues. This is in 1660. What Francesco Redi did, he demonstrated that maggots resulted from flies laying eggs on rotting meat and not by spontaneous generation. He had 3 jars down here with rotten meat at the bottom. One was open, one was covered, and one had a membrane. And only the one that was open generated maggots. So they must have come from the flies. Again, this was not universally accepted for another 100 years or so. Now, in 1665, Robert Hooke advanced the cell theory of biology. He studied fungi and discovered cells. He proposed that cells are the basic unit of all living things, beginning of the cell theory that all living creatures are composed of cells. Again, if you think back, if you think this is biology 101, but I just wanted to make you aware when this actually happened, when this theory started to develop. So this is actually the fellow I always thought who invented the first microscope. It's a Dutch, Antonie van Leeuwenhoek, and he used a microscope to describe microorganisms. This is the fellow over here. He was a tailor, and he used lenses to examine clothes. So this is probably why he led his interest in lens making. He could magnify an object up to 270 times of its actual size, and he examined all kinds of things, some things you don't want to know about what he actually examined. So he was the first to discover and record microorganisms. So this was in 1674. And microorganisms is bacteria and other stuff. We're not talking about viruses or parasites, they come later. He called these tiny living organisms animalcules. Not quite sure what that means. He first described bacteria, but then also protozoan, but he did not know that those were actually protozoans. He just called them small microorganisms. All okay? Okay, this debate over spontaneous generation continues. Oops, sorry. These two fellows performed experiments involving the life developing from non-living matters. Various media grew microorganisms only when exposed to air, a source of bacteria and molds. They theorized that all life came from existing life forms, requiring some vital force, later to be shown to be oxygen. Heating flask materials only destroyed some vital force necessary for spontaneous generation. So this actually continued. So now we're getting into 18th century, 1798. Jenner, this fellow here, helped develop a vaccine for smallpox. And he did this because he noted the similarity between smallpox and cowpox. He discovered that vaccination with a live virus of cowpox protected individuals from smallpox. Can you still do this experiment today? So when do you think was the last time you could actually do an experiment like this? Let's say 1900, 1920, 1950, 1960.
In the 1960s, they were doing hepatitis studies.
So 1960s was the last time? Yep, I agree with that. So not too far ago. It's, okay, so we haven't talked about parasites yet. So it's the discovery of the malaria parasites, which was by Charles-Louis Alphonse Laveran. This was in 1880. He's a French army surgeon stationed in Algeria, and he was the first to notice parasites in the blood of a patient suffering from malaria. This was so important that actually there's a particular day associated with this discovery. It's probably the first time I saw this. And not surprisingly, he got the Nobel Prize. This is something I didn't know. This is actually I learned as well when I went through the history of microorganisms. This is 1886. This Scottish pathologist stained lymph from a skin lesion of smallpox patient and saw elementary bodies, which he thought were the spores of micrococci. That's shown down here. These were, in fact, smallpox virus particles. And he did not know this at the time, but he was the first person to see and photograph a virus. That was in 1886. So this, unfortunately, we couldn't get to work, so I'm just going to skip over this. So then there was the discovery that so the first discovery was a patient with malaria had parasites in the blood. Then obviously is how does a person get these parasites? And this was Ronald Ross, British officer in the Indian Medical Service, was the first to demonstrate that malaria parasites could be transmitted from infected patients to mosquitoes. And in further work with bird malaria, Ross showed that mosquitoes could transmit malaria parasites from bird to bird. Thus, the problem of malaria transmission was solved. Okay, the development of germ theory of disease. Who is familiar with this theory? Almost everybody? Okay, good. Developed by Louis Pasteur, and obviously developed the process now known as pasteurization, which uses moderate heat below boiling to destroy harmful microorganisms, and this process does not chemically alter the substance being pasteurized. He also worked on cholera and anthrax and rabies, so he worked on bacterial microorganisms and viruses. And he found that attenuated or weakened organisms could not cause infections, but would produce immunity. Very important discovery. Even though he worked on anthrax, he did not discover what actually caused anthrax. And that was Robert Koch. And he found that tuberculosis is caused by a bacterium. He isolated Bacillus anthracis and found that anthrax built persisting endospores, increasing its survival odds. So that has led to the germ theory of disease. But I think what he's most known for is the development of postulates establishing a specific microbe as the cause of an infectious disease. Who is familiar with these postulates? Okay. Should I test you and ask you what they are? Okay, so he established 4 criteria. The microorganism or other pathogen must be present in all cases of the disease. The pathogen can be isolated from the diseased host and grown in pure culture. The pathogen from the pure culture must cause the disease when inoculated into healthy, susceptible laboratory animal. The pathogen must be re-isolated from the new host and shown to be the same as the originally inoculated pathogen. Are there any limitations to these postulates? Precisely. It's one. Another one? Say that again?
The inoculation and in healthy donor,
Mm-hmm.
now is it could be an animal.
Could be an animal. So that's another problem because there's no animal model with several diseases. So, yep, precisely. So there have become limitations have come up that we found afterwards. So that particular bacteria cannot be grown in pure culture. Always pure is the thing because you can add feeder cells and do other tricks, but that's not a pure culture. And there's no animal model of infection for several infectious diseases. Just to keep this in mind. Walter Reed. So now we're slowly moving into the 1900s. So during the Spanish-American War, American deaths due to yellow fever were colossal. The disease also appeared to be spreading slowly northward into the continental US. So through experimental transmission to mice in 1900, he demonstrated that yellow fever was caused by a virus spread by mosquitoes. And the infection was called through eradication of mosquitoes that carried it. And so his insights helped open entire new fields of epidemiology and biomedicine. And some of you might know there's still the Walter Reed Medical Institute in DC. Introduction of chemotherapy. This happened by Paul Ehrlich in 1908. And he saw treatment for syphilis. He coined the term chemotherapy and the concept of a magic bullet. He developed salvarsan, an arsenic compound, nothing lovely, that was specific and effective against syphilis. And he's credited with the first empirical observation of the blood-brain barrier and the development of the first antibiotic drug in modern medicine. And this was, as I said, in 1908. Antibiotics. I'm pretty sure you're all familiar with this. This was Alexander Fleming, who discovered antibiotics. It was around 1930. He was a Scottish biologist and a pharmacologist. And he made this discovery while working with a culture of Staphylococcus aureus that had become contaminated with Penicillium notatum. After noting that the mold inhibited the growth of the bacteria, Fleming isolated the antibiotic penicillin. And I think we all, those of you who work in research, a lot of discoveries are done by finding a contamination, what it did. So another advice, if something doesn't work out the way you want it, or if something is contaminated, sometimes you can find good things. So don't just throw it out and start over. Think about it and look at it carefully. So he investigated positive antibacterial effect on many organisms, and it noticed that it affected bacteria and many gram-positive pathogens. So we talked about scarlet fever, pneumonia, gonorrhea, but not typhoid or paratyphoid, which are gram-negative bacteria. But another decade followed before actually the active compound was extracted and purified from penicillin by these two fellows. But antibiotics were discovered by Alexander Fleming. Viruses. I told you that this Scottish fellow actually saw viruses, but he didn't know what it was. So there was a micrococci. This Wendell Stanley, he actually crystallized tobacco mosaic virus and showed that it remains infectious, and he also got the Nobel Prize. It's the first step towards describing the molecular structure of any virus and helps to further illuminate the nature of viruses. And that was around 1930, I believe. Max Tyler, he was the first to propagate yellow fever virus in chick embryos and successfully produced an attenuated vaccine, the 17D strain. And his vaccine was so safe and effective that it's still used today. And obviously saved millions of lives. And again, he was awarded the Nobel Prize in 1951. Really a colossal discovery. Now we're in 1952, and as I said, these are really selected historical events. If I were to go through everything, we probably would be here for a whole week. So these are things I thought they were interesting. I thought that you might enjoy hearing and just refresh your memory when these things happened. Jonas Salk killed the polio virus, but kept it intact enough to trigger the necessary immune response. And just to give you some numbers, in 1916, polio left about 6,000 people dead and 27,000 paralyzed in the United States. And in 1952, 57,000 cases were recorded in the US. And after 1955, when the vaccine became available, polio cases dropped by 85 to 90% in only 2 years. So obviously a really great discovery and application. How are we doing with questions? Doing this at the end or? Okay. Yeah, I was just reading up on the whole field of polio research and developing of vaccines. It wasn't the most collegial field, I got the sense. Nobody liked each other because there was a lot of prestige involved developing this vaccine. And actually the person I have my professorship named after, John Roman Paul, he worked in this field as well. He wasn't a big player in the field, but so were others not, but very contentious and very, apparently this, Jonas Salk wasn't the nicest person either, but that happens. So, 1970, Howard Temin and David Baltimore independently discovered reverse transcriptase in retroviruses. Great discovery. Established a pathway for genetic information flow from RNA to DNA, refuting the so-called central dogma of molecular biology. You only go one way, but not come back. So enormous discovery. Then Peter Doherty and Rolf Zinkernagel demonstrate the basis of antigenic recognition by the cellular immune system. Lymphocyte recognize both virus antigens and major histocompatibility antigens in order to kill virus infected cells, establish the specificity of the cellular immune system. So now we're getting to our age in 1973. No, I'm not yet in graduate school. I'm still doing my undergrad studies. Now I'm graduate school. So this was 83. Luc Montagnier and Robert Gallo announced the discovery of HIV, causation of AIDS. And this actually would have been pretty quick. This was only 2 years since the start of the AIDS epidemic. The agent responsible for it has been identified. It's really great progress in science. So I'm sort of finishing up this historical part with the year 2001, which when the working draft of the human genome sequence was published. Anybody remember this? Remember the history behind it? Again, two very strong-willed scientists were fighting for this, and luckily enough, they got together and decided to, well, they said they're going to publish together, but you know, nature comes out the day before science. So obviously there was a fight who goes into nature and who goes into science. But great discovery or great publications, a great step forward. But also I would like to remind you two words here, working draft of the human genome sequence. And still to date, it's a draft. I don't think that any genome sequence of a eukaryotic organism, probably even include some prokaryotic organisms, is ever finished or has ever been finished. It's still a work in progress. Even the yeast genome, which was the first one to be declared completely sequenced, even today, there's still some gaps and people adding on sequences. So keep that in mind, it's a work in draft. What was interesting, and coming back to the discovery of reverse transcriptase, is that about 11% of the human genome is composed of retrovirus-like retrotransposon, which are transposable elements in which transposition involves a process of reverse transcription with an RNA intermediate similar to that of a retrovirus. So like implicating that retrovirus was shaping the human genome. Still something we're learning how this actually is happening. Okay. So that was my personal historical perspective of microbial pathogenesis. As you notice, I stopped at 2001, because from that time on, discoveries sort of come every other day or every week. So it would have been difficult, but I hope you enjoyed going back in history many, even thousands of years, and learning about when things were discovered and how people were doing it. The movie that I wasn't able to play actually was produced by the Wellcome Trust, and it's freely available on their website for download. And it describes in a very nice way the discovery of African trypanosomes causing sleeping sickness in Africa, in Zululand. And I really encourage you to go to that website and look at it, and the way a single person, that was David Bruce, again as a major in the British Army in the Medical Corps, was doing his work in Zululand in South Africa, and that's where he established that these trypanosomes cause African sleeping sickness, and he also established that they're transmitted by the insect vector, the tsetse fly. And he did his work with a very primitive microscope, single tube microscope, and with the help of a lot of dogs. And he used dogs as animals to incubate blood he isolated from different wild animals, and then to show that if he injected this blood into dogs, they would cause disease, and he could find the parasites in there. You seem puzzled, Nancy. Yes. And there's actually, this movie is nice. There's a group of people going out to the field into a valley with about 50 dogs, and they just let them roam around. And then they come back to his place where he had his laboratory, which was a veranda, you know, open air. And he would just sample the dogs and then see whether they came back with parasites in the blood.
That concept of sentinels has been used for arboviruses too.
Yep.
Hamsters in trees and things like that, which identify previously unknown arboviruses.
Precisely. Yeah, it was, in our days, you probably would say sort of primitive, but that's the best you could do. You just go out with a flock of dogs into a field where there's supposed to be the disease, but nobody has seen it. And then they get infected, and he could transmit it and study it. He was also helped by his wife, who was generated absolutely fantastic, beautiful drawings of the parasites they saw in the blood of these different animals. So very nice collaboration. Okay. General concepts of microbial pathogenesis. mainly geared toward bacteria. It's probably something I'm not so familiar with, and I thought you might not be so familiar with. But some of the general concepts that I will go through and terminology comes out with parasite infections, comes out with virus infections. It's very similar. But obviously, you know, they all use different paths to enter cells, to establish infection. So it's sometimes difficult to sort of come up with a really generalized theme. At least I found it very difficult. All familiar with viruses? Terms you will hear, you know, the capsid, there's reverse transcriptase, has either DNA or RNA as a genetic material, has a lipid envelope, just very general terms that I'm pretty sure most of you are familiar with. Bacteria, different forms and shapes. They have flagellum, one or several. They have pili, can vary in numbers. Then a cytoplasm, and obviously they don't have a nucleus. And so the very interesting structures. Protozoans. Again, very, very different shapes. And not just between different parasites, but within parasites as well, with particular species. So this just illustrates this. This is a male Gametocystoplasmodium, and this is a sporozoite. And I very often use this when I teach parasitology at Yale. So there is a quiz to ask, what parasite is this? And this always causes problems, as Becky can attest to, because it's almost mind-boggling that this is the same parasite, especially something like Plasmodium that one is very familiar with. Very complex life cycles, just as a brief introduction. So, some terms, and I apologize if for some of you this is really very elementary, and but I thought for some of you it might not be, so it might be helpful to just refresh the memory. Virulence is the degree of pathogenicity of an organism, and pathogenicity is an ability to cause disease. And we can sort of break down path of pathogenicity that organism has to enter cells, and as this course will focus mainly on intracellular pathogens. So obviously then pathogen has to enter cells. Portal of entry, we then depend the number of invading microbes. Obviously, if you want to enter, so you have to adhere, you have to penetrate, and then evade the host immune defense. Most often you damage the host cells through different ways. And very often you want to leave the host cell, sometimes because you damage the host cell and it's enough, I'm leaving here, there's no more nutrients here. So that portal of exit. So I'm going to go through some of these, not all of them in very much detail, but some of them that I think might be important to give you a little bit of background for future lectures that will come up. So the key traits for a pathogen is the ability to adhere to a host surface and not to be washed off. I'm not sure washed off is the right term here, but adhering to the host surface, because otherwise, how are you going to enter the cells? Avoid phagocytosis, prevent host defense from destroying you, because you, after all, you want to get in there, you want to live, and you want to propagate. And you have to, obviously, you have to penetrate. You have to get into the host, and then you have to spread. And there are many different ways how to penetrate and get into a host, and I think in the future lectures you will hear much more about this. To produce enzymes, because you spread, you want to prevent host defense and cause damage at or near the site of infection. So obviously you need to produce something that can help you to damage the host. Could be enzymes, could be toxins, and I'll talk about toxins a little bit more. That's down here. Produce toxin, cause damage at distant sites. Portal of entry. So where does a pathogen enter a cell to become an intracellular pathogen? And this is the very specific routes by which a particular pathogen gains access. And actually, most microorganisms cannot penetrate intact skin. Actually, intact skin is an excellent defense. You can put a lot of pathogens on your skin, they don't go anywhere. They can't get in, they can't cause any damage. Of course, if you cut yourself accidentally or by other means, then it happens that the pathogen is there, then it can get into you and into the bloodstream. The most common portal of entry, and again, this is more focused on bacterial pathogens, is the respiratory tract. So you inhale, you know, you've all been on airplanes and you come back sick. That's when you get all this inhaling of moisture or dust particles. And it's obviously something that has to be there. It's not something that comes from non-living matter. So I think that debate has been resolved. Gastrointestinal tract, you know, food, water, contaminated fingers, and others. And then there's the parenteral route, and microbes are deposited directly into the tissue beneath the skin and mucous membranes, and this can be by bites by insects, injections, or other wounds. So parasite or organism can be deposited, and you feel something, you scratch yourself, you make a little pore, and the organism gets into you. And preferred portal event, many microorganisms can cause infections only when they gain access through their specific portal of entry. Two examples here. Streptococcus pneumonia, if inhaled, can cause pneumonia. If this enters through the GI tract, no disease is caused. So very specific portal of entry. Similar, Salmonella typhi, if enters the GI tract, can cause typhoid fever. If you have it on the skin, don't get the disease. So again, very specific portal of entry that these pathogens need to enter cells and cause the disease. And this just shows the two organisms I was just referring to. Now, I mentioned the number of invading microbes. And the two terms that you have come and will come across, ID50, number of microbes that will cause an infection in 50% of inoculated test animals. So this is the infectious dose. LD50, number of microbes that will kill 50% of inoculated test animals. That's the lethal dose. And the huge difference is in the ID50, and this is just one example, the anthrax bacilla. So if the portal of entry is skin, the ID50 is 10 to 50 endospores. If you inhale it, 10,000 to 20,000 endospores to get the same ID50. If you ingest it, 250,000 to 1 million endospores. So I'm not saying that you should ingest anthrax, but just again to illustrate that depending where a pathogen comes in, it's very, very different whether it can cause disease or not, or what the ID50 is. And this is just one example that I put up here just to make this very clear. Talked about adherence. So pathogen wants to enter cells, obviously has to adhere. And several pathogens, including viruses and protozoan, have developed very sophisticated strategies to adhere to cells, to only certain cells, or to all cells they can find, like toxoplasma, and then ways to get into cells, either using a system that the cell already has or developing new systems to enter cells. And that's establishment of pathogen in your portal of entry. Adherence or attachment is often an essential step in bacterial pathogenesis required for colonization of a new host. So that requires the participation of two factors, a receptor and a ligand. So it involves complementary chemical interaction between the host cells or tissue surface and the bacterial surface. And that's just illustrated down here, that you have a ligand that interacts with the receptor on the target cell. That's where it adheres and then can enter these cells very specifically. Okay, talk about damage to host cells, toxin production. And I know in future lectures this will come up in several of the toxins, because we were able to recruit some of the experts in these fields that will lecture to you in upcoming lectures. So toxin, poisonous substance produced by microorganisms. And two general categories, the exotoxins and endotoxins. And I think from the cartoon here, you can see how they differ. The exotoxins are proteins produced inside pathogenic bacteria, most commonly gram-positive bacteria, as part of their growth and metabolism. And these then secreted into the surrounding medium during log phase. Then they cause damage. Endotoxins are the lipid portions of lipopolysaccharides, or LPSs, part of the outer membrane, and they're generally on gram-negative bacteria. And the endotoxin are liberated when the bacteria die and the cell wall breaks apart. Some more information about exotoxins. So most genes that code for exotoxins are located on plasmids or phages. And that's a very common strategy of bacteria, that they actually have virulence factors and toxins encoded on plasmids or phages that they harbor. As I said, they're mostly gram-positive. They're byproducts of growing cells. And LD50 is smalls, meaning they're very potent. So for instance, 1 milligram of Clostridium botulinum or toxin can kill 1 million guinea pigs. Extremely toxic. And there are three types of exotoxins. The ones that kill cells, the ones that interfere with normal nerve impulses, these are neurotoxins. And then enterotoxins, they affect cells lining the GI tract. And many of these exotoxins actually have an A and B subunit. It's also sometimes referred to as a type 3 toxin. And the A subunit is the active subunit, and the B is the binding, the one that binds to the cell that is the toxin is targeting. And you will hear much more about this in future lectures. I just wanted to give you a little bit of background. As I said, the endotoxins are lipopolysaccharides of gram-negative bacteria. They may be released when cells are killed by antibiotics, and they cause fever and shock. And they also may allow the bacteria to cross the blood-brain barrier. Always this may, because this is not a generalized concept, but for many, many gram-negative bacteria, this is true. But as you know, in biology, you can't always say they all do this. But it's something that is very common, that they cross the blood-brain barrier. Portals of exit. So pathogens come in and they want to get out. Sometimes they simply lyse cells, they're out, but sometimes they go out through particular exit routes. And they also have a definite portal of exit where microbes leave the body, how they spread through the population. Again, respiratory tract, gastrointestinal, and urinary tract. And of course, arthropods and syringes provide a portal of exit for microbes in the blood. So, sort of sum this up, as I said, I went through a few of these terms that start with the portal of entry, penetration, evasion, host defense. Did not talk about capsules that protect bacteria from the host defense. There are many other things that happen here. The damage to host cells, I concentrate mainly on toxins, but there are other ways that pathogens can damage host cells and then they leave the cells. Viruses, they gain access to host cells because they have attachment sites for receptors on the host cell. And they avoid the host immune response by growing inside cells. And some of them cause cell death, other damage, but do not cause death. But there are other visible signs of viral infections, and they're called cytopathic effects. And that can include that cell cycle is arrested, release of lysosomal enzymes, they transform cells, very common that this happens. abnormal cells that have lost contact inhibition, formation of inclusion bodies, and so on. Pathogenic properties of protozoa. Obviously, they can cause damage to host tissue. The metabolic waste products of the parasite can produce disease symptoms. And some protozoa can change the surface antigens while growing in a host, thus avoiding destruction of the host immune system. And that is known as antigenic variation. And I'm sure you all have heard about this. Okay. So that's what I thought I could give you as a sort of an introduction to this course. And I hope that this course and the lectures that you will get will really help you to better understand how pathogens get into cells, how they induce disease, and what I haven't talked about, what can we do to actually prevent this and attack these. That is something still extremely challenging. We don't have all the solutions, but I really hope you enjoy this course. And I think that's it. Sure.
Good question and answer. Thank you very much. So, Chris, thank you very much for this opening lecture. We have space for questions, comments, discussion. So I'm going to open the space for questions. Anyone from here wants to start up with questions? Okay. You have to have no one here.
So your perspective, I think, historical perspective was kind of a good nostalgic visit to past times, and there's so much more that you know, when you read history, like you say, you always discover so many things that contemporary literature just passes over. And sometimes when you publish a paper, they only want literature from the past 5 years or something, and it's kind of unfortunate because a lot of the observations made historically were so thoughtful about the entire context of the microbial infection, not just some very specific small aspect of what was going on during the infection. And I think it's important to not to forget that some of the major discoveries that you went over were precisely that, you know, trying to discover what was the causal, or you know, what was causing this, you know. I remember with HIV, people had no idea. I was like, what's causing this disease? And people were making all kinds of speculation. Was it a fungus? So many different things. And being able to reach and demonstrate using postulates, if you have the appropriate access to models, is really challenging and fundamental. What I wanted to mention was that your emphasis was very much on the microbial pathogen, of course. And one of the things that people who are leishmaniacs, and probably the ones who study these African trypanosomes too, like you, is, you know, the provocation, the toxin. There's not exactly a toxin that they produce that poisons you, but something that's the host response, and it's very diverse as the human population. And I wonder if you might mention in some way, because the pathogenesis is not necessarily confined to the pathogenicity characteristics per se of the pathogen. Maybe you can give us your insight there.
There is actually one small story about the history I want to go in first, and that's protozoan parasites. So And it's a story about David Bruce. So he studied African trypanosomiasis, and he studied the development of the trypanosome in the insect vector, the tsetse fly. And he described that in 1900 at an utmost detail, how long it takes until, you know, the insect bites an infected host, parasites are taken up, then it says 18 hours later they lost their infectivity, then most parasites disappear, and then they develop and they move along to the salivary glands and they regain infectivity after 14 to 28 days. That was 1900, and up to now we haven't learned much more about how that actually works. So just to emphasize again, it's fascinating. And I went back and I was like, this is exactly how it works. And I was flabbergasted how we could actually figure this out at that time, to the point of giving us a time course of what's happening. So that's just another one of those great stories. Why did I focus on bacterial pathogens? Because it's something I don't know much about. I wanted to learn more, but obviously, you know, with parasitic parasites, it's different. Probably the most important difference in my perspective is that parasites don't really want the host to die. They want the host to survive as long as possible. Eventually they might die, but most parasites have evolved strategies that keeps the host they infect alive for as long as possible, or even longer. So that is a big difference to some, like viral infections and others. So, and to understand how that works, still extremely challenging.
Thank you. Before going to the chat, can I just ask one question, maybe touching on the subject of the host pathogen relationship? So if we could revive a little bit the spontaneous generation story debate, let's say. No. It's a big discussion about what's a pathogen and what's not a pathogen. So in many instances, commensal organisms can cause disease. And I think it's a philosophical discussion whether to call these pathogens or not. So in that context is where also host responses, let's say, play an important role in facilitating the development of disease. So I was wondering whether you can give us some comments on this issue of commensals being pathogenic, whether we should consider them a pathogen or not a pathogen. Just open the discussion. No comment.
I'm not going to go there.
No, he's not. No. Okay, has refused to go there. So
I would say something about that, because so many microbial organisms are opportunistic. And under those circumstances, they can kill you. You know, I mean, this was the thing with HIV. People were dying of all kinds of things. And so what's the cause? You know, it's like it could be 10 different things that's causing HIV, because people were dying of so many different things that were actually opportunistic. And so even a situation where Pseudomonas, you know, it's a nightmare, but it's a nightmare for those people who have cystic fibrosis. And so it's very circumstantial, I think, and as the population expands in the world and the different living conditions that create a huge selective pressure on human populations, make them more vulnerable to things that maybe weren't such a problem before. I was just reading this morning that on some big competition in London that a lot of the elite athletes got norovirus.
Yep.
You know, I mean, that norovirus, that is a nightmare. That's the cruise ship, you know, virus that, yeah, did you? Oh my gosh. I'm like, it hasn't happened here, I think, in Colombia. You haven't heard anything about norovirus, but it'll get here sooner or later. Anyway, so yeah, I think that the possibility of organisms that are even, let's say, commensals in the environment that they can actually like the Legionnaire's disease, for example. I mean, that was an example of an organism that was in the air conditioning units in a very old and prestigious hotel in Philadelphia. And the people were there at a convention, and in the rooms they got exposed. These were older, you know, like veterans, I think, at a convention, and a lot of them got exposed to this in the air conditioning within the hotel. So it wasn't really that an organism that was that. It was just discovered in that outbreak. And so I think circumstances are going to reveal to us new microbial organisms that can be extremely devastating.
Thank you. Let's go to the chat. So our colleagues at Universidad de los Andes have a question. It says, it was previously mentioned that endotoxins may be released when cells are killed by antibiotics. How does this phenomenon might limit the doses of an antibiotic therapy in a real case?
Oh, antibiotic therapy. Oh my God. Where do we start? It's probably, I think, I don't want to blame the medical community, but this is probably the most contentious area in medical doctors ever can talk about antibiotic treatment and or therapy. Because, are there any MDs here? Would you agree that it's sort of like, let's try this and see how it works and then try something else? Yep.
Well, I was thinking that we have to give the antibiotic because if we don't give it, those toxins are going to get released at any time, and it could be worse because the bacteria are going to grow even more and it's going to be more pathogenic. So I think you have to think about the patient, about the risk, and I prefer to have a toxic shock syndrome than a dead person.
Okay. Yeah.
What about prions on the discussion on what's a pathogen?
Prions. So do they exist? Well, there were long debates whether they exist or not. The question will be whether they're pathogens or not.
I guess so, yeah.
Depends on your definition of a pathogen. Would I consider it a pathogen? Sure.
They're pathogenic protein.
Yeah.
Okay. There's another
Pathogen?
question.
Thanks. So I know you might not be able to answer this, but I was intrigued because a lot of this introduction focused a lot more on the molecular aspects and differences between viruses, parasites, and bacteria. And being an engineer, I'm very interested more in the biophysics and the biomechanics behind potentially the differences between different bacteria as well as different parasites. And I was reminding myself as to which disease it was, but I think last October they found out that schistosomiasis actually uses really fascinating biomechanics in order to swim. And so I was wondering if there is any discussion or thought about further delineating different parasites or different bacteria solely based on kind of more the biomechanics or biophysical questions rather than just, releasing different endotoxins or whatnot.
Absolutely. So obviously very familiar with studies with African trypanosomes. That there's a group in Germany that looks at how they swim in blood as compared to artificial media we have, and how that relates to infectivity and propagation and changing surface coat. There are several people doing this, yes. And I obviously am not a biophysicist, so I don't know all the details, but the fascinating stuff coming. They're doing things very differently. And they're also looking at mutants, you know, that are effective in certain aspects of invasion. So how do they propagate? Not just, you know, we always would just look at the microscope and say, yeah, this parasite is a little sluggish, but they're doing it now with biophysical techniques. And yes.
Do you have other questions here? Discussion, something? Do we have any other questions in the chat, or some points of discussion, some philosophical questions? No? All right, so I think then we're going to leave it at that. Once again, thank you all for participating in the course. We're going to continue with the introductory lectures in the next session. It's going to be an introductory lecture on signaling, and that again to prepare the ground for the talks that we're going to have later on host microbe communication as well. So feel free to write emails if you have questions that pop up later in the evening or at night. We will be happy to answer them as well through email, and we'll see you then next week. So Dr. Shuri, thank you so much again for this talk, and thank you all.